Hi, and welcome to this first video for the Systematics course, formerly entitled The Diversity and Evolution of Insects. As I've recorded these videos, I made a conscious choice not to do a lot of these far in advance, just far enough in advance to get them posted to you on time, with the idea that I wanted to be able to adjust as we go along to your needs, your questions, and your input on the videos. I'm not a professional video producer, that's for sure. Although over the past year, you've kind of been wrangled into such a role. But I hope that these will work out well for you, and we can use our Thursday office hour time to address questions as we go along. So the topic for this first video is, really a very fundamental question. Maybe it's too simple a question. It seems like an obvious question, but how many species of insects are actually out there? And more importantly, I guess, how do we know? So I've assigned a couple of readings for this video. One written by Irwin a good while ago now, in 1982. And I'm going to talk about that first here. And then a follow-up, one of many follow-ups to Terry Irwin's original publication by Nigel Stork. I hope that you have read these in advance. I don't think it's going to be a deal breaker if you haven't read them yet. But some of them, what I'm going to talk about might make more sense if you've already tried to work your way through and think about what they're talking about. So let's take a look. Both of these papers deal with ultimately the question of how many species of insects, or more generally, how many species of arthropods there are in the world. So before we get into the details of those two papers, we need to ask ourselves some questions. First of all, why the heck should we care how many species there are, or do we care at all? There are even the smallest estimates of the number of insects that are out there puts the number in the hundreds of thousands. There is no person on this earth now, or ever was, or ever shall be, who had the ability to identify, to recognize each and every species of insects on the earth. You may have some people with extraordinary capabilities that can recognize all the birds in the world, or maybe all the mammals in the world. But those groups of organisms, the total number of species, you're talking in, well, depends on which species concept you talk about, and that the idea of species concepts and what is a species at all is something we're going to get into in the next video. So just leave that question in abeyance for the time being. But what are there? Roughly, I've forgotten the exact numbers, something like 10,000 species of mammals. And the way I learned it way back in the day was that there were approximately 8,600 species of birds. I think maybe the idea now is that we might have double that number of species, simply because we recognize things that used to be called species at the higher level now. But even so, that is less than 10%. Maybe even it's only 1% of the total number of insect species in the world. So we're talking about an order of magnitude more. So if we can't identify them, and there's so many, do we really care if we have delimited each and every species and provided a means for its identification? I would submit to you that the answer is yes. Of course, I'm prejudiced because that's what I do for my research. One of the foci of my research is to look at the parasitic wasps we study and try to figure out what all the species are and find ways to distinguish them. So obviously, I'm coming at it from a prejudice point of view, but let me give you some of the initial ideas that come to my mind about why we should care about how many species there are. So every species, ecological theory tells us that each and every species is distinct in some way. They represent distinct historical entities who have evolved in isolation from other such entities for varying periods of time, but maybe dozens of years in extreme cases of rapid speciation, thousands of years, millions of years, tens of millions of years in some cases. So if each and every one of them is distinct, then their biology is distinct. What they do and how they do it is different. And in many ways, we care what insects do. If we're going to effectively control things like, here's my list, pests of food crops in order to assure a food supply, pests of forests so that we have access to building materials, to paper. Maybe in this electronic world, we won't need so much paper, but paper, building materials, what else associated with forest pests? Oxygen, hydrologic, the components of the water cycle, all of these things that we care about the existence of forests. Oh, fuel, how could I forget fuel? The burning of wood for fuel is still a very, very important thing in many parts of the world. There are many medical and veterinary pests where we care about what the different species do. One of the other courses I teach focuses on malaria. And there are, although all the vectors of human malaria are species in the genus Anopheles, many of those species differ in some very important biological characteristics, whether or not they are capable or willing to feed indoors. What time of day do they feed? How much do they prefer to feed upon humans than others? Get the species that differ. And if we can't tell the species apart, then we can't adequately develop the control methods that we need in order to control the mosquitoes. It doesn't do any good to have indoor residual spraying to kill malaria-bearing mosquitoes if those mosquitoes are not living indoors, but they're actually living outside. So if it's important for us to know the biology of the individual species, in order to do that, we need to know what those species are and how to recognize them. Well, that's the negative side of insects. There's also a very positive side of insects, and the same thing follows. There are many natural enemies of insects. And if our desire is to foster, conserve, and help to those insects to do the things that they do in the world, to prey on or to parasitize other insects that are economically important or important in food or important in health, then we need to know what those species are, how we distinguish them, whether or not we can import a natural enemy from, let's say, southern China into the northeastern United States and have any expectation that it's going to be able to control the pest species that it's brought in to control. So the same basic argument, just taking the opposite point of view. If insects do bad things, then we need to know the details of their biology in order to help control them. If insects do good things, then we need to know the details of their biology in order to facilitate them doing that. Let's see, what else do I have here? Indicators of habitat quality. Insects can be used as a species in order to assess environmental degradation or restoration, if that's what you're interested in. I put the metaphor, I think it's a metaphor, of the canary in the coal mine, that if we see certain species disappearing, that tells us something about the quality of the environment. And finally, maybe this is not something that is important to everyone, but certainly I find many insects to be things of absolute beauty. Maybe we should have a poll sometime on what everyone's favorite insect is. I'll bet you'll never guess what my favorite insect is. But they can be things of beauty, things of amazement, things that make the world a better place for people. And at the same time, you may have ethical concerns about habitat destruction and associated species loss with extinction and things like this. And so it's impossible to conserve what you don't know is there. And so this is just a preliminary list that I put together of some of the reasons why we might care to know what all the species of insects in the world are. So then moving on, if the basic question is how many species of insects are there, there's actually two components of that. One of them is, well, how many do we know about? And it may surprise you to know that there is no authoritative source or a reference to tell us exactly how many insect species have been described and named since 1758, the starting point for zoological nomenclature. We don't know. Estimates are, well, somewhere in the range of 750,000 to maybe a million. That's a big ballpark. And it's only in the case of some very, very small groups that we have really a total global understanding of how many species have been described. Add to that there are many species that we don't know about. It might sound contradictory to say we know that we don't know, but every year there are thousands of new species that are described and named and labeled as quote unquote new to science. How big is that piece of the pie? How does it compare to the number of species that we already know about? Itself an unknown number. Okay, as I said, number of described species somewhere in the area of 750,000 to a million for large groups like beetles, for the Hymenoptera that I study, for Lepidoptera. Lepidoptera is probably in a fairly better position just because of the large size and attractiveness of many butterflies, but that leaves all those micro moths, doesn't it? There's simply no authoritative source for what has been actually published in the world. That's one of the things that I've been working on for I hate to admit it, but decades, and I'm still whittling away at is probably a better word, to come up with a complete survey of what has been published. Estimates, and I say estimates because again, nobody has documented all of this. Somewhere on the order of 10,000 new species are described every year. That is things that people have discovered and said, there's nothing like this in the literature that has been recorded before. I'm going to call it a new species. And this number is, fairly steady. It depends, of course, on the number of people actually trying to do the work, and that's an issue that we'll definitely get to toward the end of the course. But that's something that weighs on my mind quite heavily. How many taxonomists are there in the world that are doing this kind of work, and what are the priorities for the work that they do? But I guess I should also tell you what I mean by description. About halfway through the course, I think we're going to talk about the rules of zoological nomenclature. But for the time being, when I talk about a description of a species or described species, that means a species or any other taxon, any other level in the taxonomic hierarchy, that somebody says this thing, this set of specimens, this set of individuals are a group that has never been recognized before, and I wish to delimit it and give it a name. And the delimitation part is an essential part. It doesn't do much good to say, oh, I saw a new species the other day and I'm going to name it Dibubliomuctus flavipes. If you see it, let me know if I don't tell you what it is. And so the delimitation is the description, the actual giving of how you think the species, in this case, that you think is new, how other people can recognize it and distinguish it from other existing species. And in order for a name to be recognized under our existing rules as a validly proposed name, it has to be accompanied by this description and then published, either in hard copy There are now means of doing this in electronic format. But it has to be put out there for the rest of the world to see and to comment on. Doesn't mean it has to be right, but it has to be published in some sense. So when I say new species described, I'm talking about these are basically hypotheses of what's out there in nature that have been put in the scientific literature for everyone to see, comment on, and test. So 10,000 new species, roughly, described each and every year. And as far as I can see, there's no ebbing of that flow. And this is a guesstimate, even in these days of extensive indexing of the published scientific literature. Taxonomic works are published in literally thousands of journals around the world, big and small. Some the organs of large societies, such as the Entomological Society of America, the Royal Entomological Society. Others from minute little groups. Even some, it's still valid for people to publish these descriptions in essentially the vanity press, to go and pay to have something. Well, we pay page charges, don't we? But to actually go write up your work, take it to a publisher and say, please publish this under my name, and I'm going to give it a new name and pay you the money to do it. You could go to Amazon and publish what under the existing rules we would have to accept as valid publications through their publication services, which are just vanity presses. I will get into all those gripes and criticisms when we get to talking about the rules of nomenclature. So let me grab I'm going to try to grab my pointer here so that I can use it as we go forward. Okay, there we go. So let's talk about these two papers a little bit that purport to describe the number, not describe, but purport to estimate the number of species of insects and arthropods that there are in the world. The first one was authored by Terry Irwin. As you can see from those dates, Terry just passed away last year. The word that I heard that has not been verified, not that I've sought verification, was that he was a victim of the COVID pandemic. If you do the arithmetic there, you'll see that Terry was 79 years old. He hadn't quite reached his 80th birthday yet when he died. But he always, that picture, I don't know when this image was taken, but this is how Terry looked up until the last time I saw him, which was about a year or so ago. And he was a regular attendee at the meetings of the Entomological Society of America. A very boyish grin on his face, graying only at the temples, big full head of hair. I assume it's real hair. I never asked him. But he was an active member of the Entomological Society of America and well respected and well known throughout the community. He worked at the Smithsonian Institution and the Natural History Museum there on Constitution Avenue, and he was a specialist in ground beetles, although that's a bit of a misnomer because many of the beetles that he specialized, for example, that image on the right-hand side there, that genus is called Agra, A-G-R-A. These are arboreal beetles. These are beetles that, I haven't looked into what their immature biology is, but as adults, these are predators up in the forest canopy. And of course, it's the forest canopy that was the focus of the paper that I have assigned for reading for this first class. So if you've never been in a tropical forest, let me quickly describe what the overall structure of the forest is. So whereas in like the deciduous forest here in Ohio, basically you have a set of trees, and the trees are generally more or less the same height, 1 to 200 feet. Well, probably even less than that, 100 to 150 feet, maybe down to 75. But you get a layer of trees that form the canopy, the top level of the forest. And then down underneath that, depending on how much light comes through that canopy, you could have an understory, or as this image puts it, the under canopy of smaller trees. In Ohio, you know, the canopy might be made up of oak trees, maple trees, hickories, and things like that. And then in the understory, you get smaller trees. You get dogwoods and sassafras and redbud, you know, things that don't grow very tall, um, forming that substratum in the forest. And then down on the forest floor, you have, well, they're calling it here shrubs, but it doesn't necessarily have to be those types of plants. You could have some shrubs, things like viburnum or ferns, plants at the ground level, trillium, wild ginger, mayapple, jack-in-the-pulpit, these kinds of low plants. So basically you have three layers. You have the upper canopy, a subcanopy or under canopy, and then the herbaceous layer down at the bottom. In a wet tropical forest, though, it's a little bit more complicated. We have, as it shows here in this illustration, we have a canopy of trees that are more or less, you know, roughly the same height. But above that, there are certain species of trees that called emergents that actually grow significantly taller than most of the trees in the forest. And so these trees really stand out as the high points. You can have an under canopy, but because this layer here in the canopy is typically so dense that very little light gets through to these lower layers. In the kind of wet tropical forest that I've been in, the area down here, the shrub layer, is really very sparse. There's not a lot of vegetation going on down here. So in contrast to the when I was growing up, I always loved to watch the Tarzan movies with Johnny Weissmuller and the sort in the jungle of Africa, and see them with the machetes whacking their way through the dense, thick forest. You're only going to get those lower dense layers in areas where there's enough light to support those plants to grow. So in a typical tropical forest, in fact, it's more like an open parkland where you can walk easily between the trees. And so you have very little in this lower layer down here. So with that said, here is an image of a typical tropical wet forest where you have, well, at least the top of it, where you can see how we have this layer of trees in the canopy and then a few emergents that rise significantly above those areas. Obviously, we can't see down here below as to what's going on underneath in the subcanopy or the shrub layer, herbaceous layer below it. I wanted to show you this picture because you can go to some places. This is a place called Reserva Duque, which is a reserve in adjacent to the city. You can see the city there in the background of the city of Manaus in central Brazil in the central Amazonian rainforest. And at Duque was an entomologist, Adolfo Duque, who worked in Brazil in early part of the 20th century, I think, late 19th century, early part of the 20th century. Anyway, I put this picture in because at the reserve, they have one of these towers that you can climb up in order to go through from ground level all the way up and above the forest canopy and look out above there. And up there, they have meteorological instrumentation. You can see in order to get there, you see these are the stairs that you go up and up, back and forth to go up. I tried to climb this tower. And I want to tell you, you can't see in the picture there, but there are guy wires that are holding this thing because it's going up like, you know, I didn't look up the exact height, but a couple of 100 feet, maybe even 300 feet into the air. So the height of a football field up into the air. Once you get up about, I'd say halfway, you can start to feel that this tower is kind of moving back and forth. Get up 2/3 of the way, and the tower is moving a little bit more energetically. I think I made it up about to there before my gut just couldn't handle it anymore, and I had to say, I can see the top from here. Good enough. I'm not heading up any higher because I wasn't getting seasick, but my knees were getting pretty weak. I'm not ashamed to admit it. There was another reserve we visited during this trip that had a much more substantial tower that goes up through the canopy. Same height, but it was about at least twice as wide as this one and made of much sturdier materials that I got up without any problem. I mean, yeah, you could feel a little bit of shimmy shake up toward the top, but nothing that really would have all of the gumption drain right out of you as you climbed up. But that just gives you an idea of what a tropical forest looks like if you've never had the pleasure of going there yet. We'll say yet, right? Every biologist should definitely visit a tropical forest at some point in their lives. So, in contrast to this wet tropical forest in the middle of the Amazonian jungle, the habitat that Terry was working in the course of the paper that he talked about was a less well-developed, he even called it a scrubby seasonal tropical forest in Panama, in the canal. I believe it was all done in the canal zone, or what was then the canal zone. Now it's all Panama, of course. And the focus of the study, the studies that preceded that paper, were to sample the beetle fauna of this little tree, Luea seymani. And so over the course of 3 years, he and colleagues tried to collect and sample and identify and count every last beetle that they would collect from, 3 individual or 19 individuals of this tree in these scrubby seasonal forests. By seasonal, I mean that there's a wet and dry season and they'll lose their leaves and are not the evergreen forests that are typical of the Amazon basin. So here we have this one species of tree in which Terry attempted to census the entire population, both citizens and non-citizens, of beetles in it. And the method that they use, and he pioneered and then extended, this is a picture of him, I believe this picture was taken in Peru, was canopy fogging. Now if you get into a serious tropical forest where the trees are hundreds of feet high, one option, and I have met people who have developed the climbing skills to actually go up those trees in order to collect insects. Not for me. I don't have those skills. We'll never have those skills, that's for sure. Even if you do that, though, you're pretty limited as to your mobility up in the forest canopy to collect. It's not like you can go with a net and go running after a particular species that you see crawling around in order to grab it. You're pretty much limited to where the ropes will let you go. So a different approach to this is what you see here called canopy fogging. Basically what you do is you come to your sampling site in the early morning before you start to get convection currents generated by the warming of the air layers by the sun. So it's early morning, and there are many varieties of these machines. Basically, this fogger is a heater in which you place a contact insecticide. That is something that if an insect comes in contact with it, they are knocked down. But one that can be volatilized, warmed and volatilized, and then here you see this warm air carrying the insecticide rising up into the canopy. It's rising because it's warm as opposed to the cooler layers of air down below. You might think a tropical forest is hot all the time. In fact, a tropical forest at night is fairly chilly. It can get down to around 60 degrees. And even during the day, down underneath in the shade, it's quite tolerable and quite pleasant. Humid, but warm. And so the warm air, and maybe there's a water base here or something, I don't know the details of the formulation here, carries the insecticide. It rises and fogs the trees that it comes in contact with. And the insects that come in contact with this insecticide basically start raining out of the treetops, falling down. They're immobilized and they just fall out, and you get this rain of insects coming down. And down at the bottom, if you're doing this quantitatively, you've arranged collectors. In this case, these are funnels. And in order to do it quantitatively, you would know the area of the funnels. And so you get X insects falling into Y square meters of area. You can calculate how many insects there were up in the tree that had been knocked down and fall out, even if you don't collect them all. And so here we see Terry here on the left. I'm not sure what they're trying to do, probably moving some big showy things or things of particular interest from these funnels, but they would collect them all, bring them back in the work they did in Manaus. They bring them back to the Institute there, the work in Peru. They do some of it locally or bring stuff back to the Smithsonian Institution to be sorted, counted, identified in order to document all of the insects that are falling out of there. So you can get both a sampling of the species diversity up in the canopy and also an idea of the numerical abundance of these insects. Surprisingly, of course, things like mantids and larger beetles and things like that, you'd expect to be up there. But other work, in particular, one similar study that took place on the other side of the world on the island of Sulawesi in Indonesia, some of the most abundant and most species-rich groups were minute parasitic Hymenoptera. I mean, when I say minute, I'm talking about things 1 millimeter, half a millimeter total body size that turned out to be incredibly species rich and incredibly abundant in the canopy, and things that experts had never seen before. These things had never been collected for. They apparently spend their entire lives, their entire evolutionary history, if you will, in the tops of the trees, never coming down to the bottom where they would be netted by a passing entomologist. So with that, what did Terry do with the data? You've read this, or you will read this, but let me just quickly go over it. So from the Luhia study in Panama, they counted the number of species of beetles that they collected, excluding weevils. Now that's a pretty darn big exclusion, since weevils are arguably, and everybody argues, the most species-rich group of beetles, and beetles were arguably, again, the most species-rich group of animals. Even so, weevils are notoriously difficult to identify. They collected 955 species of beetles other than weevils on their sampling of trees in Panama over the course of 3 years. So I guess it was 19 trees over the course of 3 years. Collected nearly 1,000 species of beetles. Mind-boggling. And this is in a scrubby, unimpressive forest. Based on other studies, Irwin estimated that if we have 1,000 species of other beetles, then weevils are so common, they make up about a quarter of all beetles, and so he estimated there were 260 species of weevils in his sample. So roughly speaking, about 1,200 species of beetles. Luhia seimanae is, of course, only one species that you would find, and one of the ways that they frame the question is how many species are living in a hectare of tropical forest. So How many species of trees are there in tropical forest? Now, trees can't run away. Trees are pretty obvious. Um, they're by definition big, so it's relatively easy, and people have done this, to go in and census survey the number of species of trees in a tropical forest, and the numbers are just astronomical, up to 204, or an estimate. A common number is hundreds of species of trees, some of them represented only by a single individual. So tremendous levels of species diversity, both in absolute numbers and in the evenness that you have. But because some species are closely related to other species, so they're not really that different, Terry pared this number eventually down to approximately 70, I think he called it genus group sorts of taxa, that these are species that are very distinctive, at least taxonomically. So 70 of these in 1 hectare of tropical forest. How many are there in temperate forest? Often you're talking single digits. Excuse me. So given that, he knows the number of beetles on 1 species of tree, and he estimates somewhere, a conservative estimate, as there's 70 different kinds of trees in a hectare. How many of those beetles are specialist herbivores on that tree? That is, they need to feed on that particular species, maybe because of the secondary chemicals in the tree, maybe because of the phenology of the tree when it fruits, when it flowers, those kinds of things. How many beetles are herbivorous and specialize on that species of tree? And again, I mean, there's some literature to guide. There was some literature to guide him on coming up with this number, but it's hardly definitive. But the number he used was 20%, 1/5 of those beetle species. He estimated were going to be specialists to depend upon that tree for their reproductive capacity. And then, of course, we have the other trophic levels. We have predators, of which parasitoids would be, well, there are a few parasitoid beetles, but not many. You got predators, lots of fungivorous beetles, those feeding on lichens and fungi and all these kinds of things. And then there's also scavengers. And so the question was, how many of those would rely upon this particular species of tree, or rely upon another organism like an herbivorous beetle that in turn relies upon the tree? So how many of these predators, fungivores, and scavengers would be reliant upon this particular species of tree? And the working numbers that he used for this brief paper were 5% of the predators were reliant on that tree species, 10% of the fungivores were reliant on that tree species, and 5% of the scavengers. So you can do the math, extrapolate from the number to figure out how many beetle species are reliant upon that tree, then multiply to how many trees are in a hectare, and then extrapolate to from the number of beetles to the number of total insects or total arthropods, if that's where you want to go. You want to include spiders and mites and things like that. And then extrapolate the canopy fauna that he has sampled to the fauna beneath the canopy. So the fauna in that subcanopy region, in the herbaceous or shrub layer, to the edaphic fauna, to the fauna in the soil itself. And then extrapolate from the single tree to all tropical trees around the world. I guess he was figuring that the temperate faunas would be just a small blip when compared to the tropical fauna. And the number he came up with pretty much not only shocked himself, but I think it was a fairly shocking number for everybody else in the world. I mean, this is a 2-page paper filled with, shall I be kind or not? I'll be kind. Filled with speculation about the percentage of herbivorous beetles, the percentage that rely upon this tree, et cetera, et cetera, et cetera, the comparison of the canopy, all this kind of stuff. These are soft numbers. But this number was so much beyond even the highest number that people had estimated before that it has grabbed it still grabs the attention of the world community. The number he came up was 30 million species of, was it insects or arthropods? I've forgotten though. Right, you'll have to look it up and tell me which one it was. At least 20 times what the highest estimate was. And so this little 2-page paper filled with lots of speculation and extrapolation is probably one of the most cited papers ever published, and in a minor journal like the Coleopterous Bulletin. I'm sorry if you're a coleopterist, they would not like me to call the organ of their society a minor journal, but let's say it's got a rather restricted readership. So that was one of the earliest estimates that really knocked us off of the comfortable position we were in about how many species of insects there were. I mean, if there's only 1.5 million species and we've described 750 or 1,000 or 1 million of them, then we're at least halfway, maybe what, 2/3, 1 150 million, 2/3 of the way to the total. In contrast, we're maybe 5%. If Terry's number were right, then in terms of the number of species that have already been described, we're only 5% of the, what is 1/30th? That's not 5%, is it? It's an even much smaller number. 1 in 20 would be 5%. 1 in 40 would be 2.5. So about 3 or 4% of the way along in this, and we've been working on it for 250 years. When would we get to an endpoint on that? Do the math. It's long beyond the time that I plan to retire. So let's move on then to briefly talk about this paper by Nigel Stork. Nigel Stork has done a lot of the similar sorts of work, but in different parts of the world, in New Guinea, for example, Australia, I believe. But this paper was not really about going out and sampling and collecting new data from canopy fogging, but this was to look at, since the time that Irwin first published this, up to the time this was published in the Proceedings of the National Academy of Sciences of the U.S. in just 5 years ago, 6 years ago, to look at different methods and the estimates they had, and to introduce a new method for estimating how many species of insects and how many species of arthropods. So of the 8 separate methods that he cites, one of them was Terry Irwin's paper, and there were some others, some more theoretical than others. But the range for the number of beetle species that he cited, the range from the minimum to the maximum of these 8 methods, was from 600,000 to over 40 million species. So that's quite a wide range. And for all arthropods, somewhere around 2.5 million up to over 80 million species of arthropods. Remember that the most diverse classes of vertebrates, things like fish, have, I think the number of fish species is somewhere in the order of, well, it's between 20 and 30,000 known species of fish. And here we have 81 million species of arthropods. Even at the low end, 2.4 million species of arthropods. But I guess the crux of the paper is that they, using all these methods and trying to compare them and put them together, basically sort of a meta study, was that the number seems to be narrowing down the range from this overall range down to somewhere in the order of 5 million species or so. They did introduce this one new way of estimating the number of species that I thought probably was a bit confusing, and so I thought I'd walk through this a little bit just to explain their method. So, let's just go back half a second. It won't allow me to go back. That's fine. Basically, what they're doing is using information on the body size of species and how and when those species were described in order to make this estimate. And a lot of the work they did was based upon the collections of what we now call the Natural History Museum, what in the days when I was a graduate student was referred to as the British Museum Natural History in parentheses. So if you've ever been to the British Museum or heard of the British Museum where the mummies are and all these cultural artifacts, that's in a different part of London. The Natural History Museum here is in South Kensington on Cromwell Road. I was there just a couple of weeks after the car crash that killed Princess Diana. Are you all watching The Crown to relive that experience? And saw all the flowers everywhere. They were in pretty decrepit shape at that time because had been a couple of weeks and they'd been sitting outdoors. But anyway, it's in that part of town, very posh part of town. And so this is the Natural History Museum. Now at the end, toward the left-hand side of the picture, they've put a new addition on called the Darwin Center, where now the work on the insect collections is focused. Very famous museum built in the 1800s. It's still there. So I digress, right? I'm going to digress a lot. So the work that Stork is, or at least the part about this body size versus year of description was based on the collections, the beetle collections of the Natural History Museum, which is one of the best. I'm sure Max Barkley, the curator there, would claim that it is the best beetle collection in the world in terms of the not just raw numbers of specimens, but in terms of its representation of the entire beetle fauna of the world. So, this is the graph. Yes, this is the graph that they published in the PNAS paper that you read, where basically what we have, let's see, on the x-axis is the year in which species were described. So it's starting in 1758 with the publication of the 10th edition of Systema Naturae. And the observation that I made before is that it seems to make sense, right? That the first things that are going to be described are the things that are most obvious, the things that are biggest, the things that are brightest, the things that are most abundant. And that as time goes on, people's attention, well, first of all, you describe all of those showboat species, but then people's attention gets focused on more narrowly and on smaller and smaller individuals of insects. I mean, the insects that I work on are in the range of 0.5 millimeter to, a big one is 10 millimeters long. That is a monster. What we see on the y-axis, the x-axis is the year in which a species was described. And on the y-axis, of all the species that were described that year, they took the mean and confidence intervals of the body size, basically the length. They were using length as a proxy for, well, it would be a proxy for the mass of the body, which I think would probably be the most important biological characteristic here. And so this is the mean body size with a natural log transform and the 95% confidence intervals. So the species that were described in 1758, this was the mean, but the 95% confidence intervals range from here to here in terms of their overall size. And we can see through time, indeed, that empirical observation that Gaston had made years before held that species that were described later tended to be smaller as we go along. Now, as Stork et al. Point out, this axis is kind of arbitrary in the sense that, maybe from 1850 to 1875, there were twice as many people working on beetles as there were from 1790 to 1810 or something like that. So the fact that the steps that are being made here are kind of arbitrary. In order to make it more meaningful, we'd have to do things of scale, like how much effort, how many people were involved in this kind of thing. So they also referred to an alternative representation of these data in a supplementary file, and I'll bet nobody downloaded that to look at it, but here it is. Basically, substituting the year of publication here with the cumulative number of species. So if you described 100 species in the first year, boom. And in the second year, 50 species were described, and this would be 150. And so just as we add them up, we're getting to the total number of described species of beetles. Here we have the same x-axis, so the body size and the 95% confidence interval. So what looks like the line, these data here are for all beetles. So then what they did was they went in and took a purportedly random sample from the beetle collection in the Natural History Museum and counted the numbers, took each species in a drawer that they randomly pull out of the collection, measured the body size, and accumulated all those data. At the end of the day, for their random sample, which would purportedly represent a random sample of all the beetles in the collection, they calculated the mean, body size, and the 95% confidence intervals for their sample. Of course, this represents a sample of all the beetles known in the world that are now represented in the Natural History collection. And they give numbers like, maybe it's roughly half of total all the beetles in the species in the world are in the Natural History Museum's collection. When they did that, then they looked at that mean and those confidence intervals and said, where on this graph do those numbers lie? And here it is right here. Boom. The numbers in their sample were up here toward the large end of the beetle size. And if you bring this over to where it intersects this, it's not a line per se, but you get what I mean, then it falls in this area that their sample, the parameters they measure for their sample, the mean body size and the 95% confidence intervals, matches what was known in the period from about 1758 to 1762. In other words, in only the very earliest times of beetle discovery and description in the world. So let's see. Their sample, of course, their size corresponds to what was known in 1758 to 1762, suggesting that many, many smaller species of beetles are still unknown, or at least unrepresented in the Natural History Museum's collection. So in 1758, we knew of 352 British species of beetles. 1762, 437 species. Now we know of over 179, nearly 180,000 species of beetles. In other words, the proportion of known British species to the total number of species now known ranged from, now there's 510 times more than this small number and 411 times more than this larger number in 1762. So that's a portion of beetle species to, the proportion of British species compared to the total number of species in that same time period. We now know that there's not 352, not 437, but approximately 4,000 species of beetles in Britain, United Kingdom. So if you then use these same multiplying factors, where did they go? Here we are, 411 to 510. That means there's somewhere in the area of 1.7 to 21 million species of beetles in the world. Again, I guess the lesson I would take from this is that the absolute numbers are not that critically important, but the lesson we must draw, I think, no matter how you, and these papers are rife with weak points susceptible to really quite severe criticism. They all have very, very, very weak points that you would say, well, I can't really trust these numbers. But I think it's important that all of these numbers no matter which ones you pick, tell us that the number of species that we know about now is a very small proportion of how many species there probably are out there. I mean, one of the things people always ask me when I talk about my little wasp and how many species there are yet to be described is, well, how do you know how many species are yet to be described if you don't know what they are? So it's a difficult question to answer, and it always involves assumptions. And the flip side of saying that there's unfounded assumptions here is that you're speculating. But all indications are that what we know about in terms of the richness of insects, the number of species of insects in the world, tells us that there is still such a tremendous amount that we still don't know. And that's about the living fauna of insects. This does not even begin to address how many species have existed through time. So, a quick summary before we close this out. Basically, I think it's safe to say that we still, quote unquote, know only a very small proportion of the total number of species. We haven't even begun to address the question of so-called cryptic species. Species that physically look the same, but if you start looking at maybe their behavior, their phenology, the molecules, what looks like one species is actually several species. I have an example of a species that in my dissertation I said was one species. We look at DNA and it tells us that, well, maybe there's not one, maybe there's 40. All looking, or at least looking to a human the same. Presumably, the insects can tell themselves apart. So, whatever these numbers that we're talking about so far, we're talking about species that are apparent, discernible to human eyes. Multiply that number by an unknown multiplier for the number of cryptic species. Is it 40? Maybe that's a bit high on average, but could easily be 2, 10, who knows? So if you accepted, or at least tolerated, my initial answers to the question of why do we care to know, then the next question is, if we do care to know, how do we make progress? If in 250 years of research, more than 250, we've only made such a small dent in elucidating the true diversity, how do we make any substantial progress in a reasonable length of time? One of the things that is inhibiting this is that in order to make progress, you have to have people working on it. If you're going to have people working on it, you have to have jobs for those people. If you're going to have jobs for those people, you have to have money to pay people, not only doing the research, but just to pay them money to eat and have families and survive in their professions. And the amount of money that is devoted to this, given all the things in the world to spend money on. Insect taxonomy is, I don't know where it is on the list. I don't know if it's rising or dropping on the list, but it's certainly not anywhere near the top of the list. That we are we have a deficit of manpower, which in part may be due to a deficit of funding. In part may be due to a deficit in stature of these positions. A lot of people think that taxonomic work is, didn't get all that done in the 19th century. This is the 21st century. Move on, man. Let's move ahead. And then finally, how do we prioritize the resources to make progress? So I guess we first have to insist that we need to make progress, and then show that we can make progress, and then perhaps we will make progress in the future. So you can look at this as a glass half empty or glass half full. I choose to look at it as a glass half full, that we have exciting times ahead of us. We have lots of new tools to discern species. We have more access, COVID notwithstanding, more access to parts of the world that before were, we couldn't get to because of disease, because of politics, because of war, transportation, all of these things. So things are looking up. I guess the real bottom line is there are a lot of species of insects out there. So with that, I'm going to stop for today, and then next time we'll talk about species concepts. Thanks.